forward mutation, reverse mutation, and neutral mutation. Forward mutation is such a mutation that converts the wild type phenotype into the mutant type phenotype. With the help of such mutations, changes occur, mean if there is a normal phenotype, that phenotype will be converted into abnormal phenotype, or we can say the functional phenotype is converted into non-functional phenotype. Same is in case of genotype. A functional genotype is converted into the non-functional genotype. Wild type is such a type in which we see that the most common phenotype which occurs in the organisms. If a phenotype is the most common in the organisms, that phenotype is called as wild type phenotype. On the other hand, a reverse mutation is such a mutation that converts the mutated phenotype into the wild type phenotype. Mean that with the help of that mutation, if there is a mutated phenotype and reverse mutation converts the mutated phenotype into the wild type phenotype. Neutral mutation is such a mutation in which we see that there is a change in the sequence of the amino acids, but we do not see the effect on the function of the protein. At the genotypic level, we see that the sequence of the amino acids has been changed, but the function of the protein remains the same, or the phenotype that remains same. Such a mutation is called as neutral mutation. Look into this diagram, we can see that what is neutral mutation. This is the genomic sequence, that is DNA sequence, and this is RNA sequence. So if we see that there is a change in the genomic sequence, instead of C there is A, the sequence has been changed, that is a codon. Because the C is converted into A, at the RNA level it is now CUU. Instead of CUU, it is AUU. CUU is the codon for amino acid leucine, while AUU is the codon that is for isoleucine. We see that in the sequence of the amino acids, in the original sequence of the amino acids, there was leucine, while as a result of the change in DNA sequence, instead of leucine, now there is isoleucine. But the function of the protein remains the same after the change of the amino acid. So if there is a change in the amino acid sequence and the function of the protein remains the same, such a mutation is called as neutral mutation. So we can conclude that forward mutation is such a mutation that converts the wild type phenotype into mutated phenotype, or we can say that functional phenotype into non-functional phenotype, while the reverse mutation converts the mutated phenotype into the normal phenotype. In case of neutral mutation, there is a change at the level of protein, but the function of the protein or the phenotype that remains same.